Dr. Pierce received his PhD in biochemistry from University of Wisconsin-Madison and his MD from Harvard Medical School. He was recruited to the Department of Ophthalmology at the University of Pennsylvania Medical School of Medicine. He returned to Harvard in 2011 to establish the Ocular Genetics Institute. Dr. Pierce's program is dedicated to identify new IRD genes, investigating the mechanism by which mutations in the identified genes lead to blindness, and using this information about disease pathogenesis to develop rational therapies to prevent vision loss. Dr. Pierce is the immediate past chair of the Foundation's Scientific Advisory Board, where he continues to serve as vice chair. Dr. K or Dr. Pierce, would you like to add anything before we begin with the question and answer session?
No, thanks for the very kind introduction, Chad.
Thank you for having us. We're excited to be here. It's always fun to do these talks.
Great, great. I think first off, I'll just sort of see the conversation with the question. Can you just give me your just very high level view, very sort of like elevator pitch answer to why is this area of research so important to you? And we'll start with Dr. K. Start with you, please.
So I did my fellowship at University of Iowa, where one of the faculty there is Ed Stone, who has always been at FFB Visions conferences. So people in the audience might have heard that name. So I just developed a love for taking care of patients with inherited retinal disease, and I find that these patients and families are extremely motivated, just very wonderful people, and I've enjoyed getting to know a lot of patients and families over the last maybe 8 years of going to Visions conferences. This is my first virtual Visions conference, so it's a little bit different, but we got to do what we got to do during COVID. But as far as why I think this is an exciting field, and specifically why I'm really excited that I got invited to the LCA Q&A today. You know, it's a rare disease, but with that said, we have multiple gene therapy and antisense oligonucleotide therapy trials that we can discuss. I'm sure we'll get questions on today. So it's a very active research field right now, and we have multiple trials that we can discuss and give you some updates on, and future updates as well. And of course, it is very exciting that we now have Luxturna, Voretigene and Parvovec, the one FDA-approved therapy for RP65 associated LCA. So, you know, compared to the whole field of inherited retinal disease is very exciting, but perhaps the most exciting field for me to be involved in as a clinician is LCA, because it kind of defines the word translational. We're finally able to bring some of the basic science advancements into the clinic, and even one of them is FDA approved and commercially available. So it's very exciting to have watched that process over the last 10 years and to be a part of it, and to watch patients having received therapy. I have 3 patients who've received Luxturna commercially, and several patients who were in the trials beforehand. I know Dr. Pierce, of course, does as well. So it's really remarkable getting to be a part of that process and watch a patient receive therapy and see the benefit. So it's been very exciting.
Thank you. Dr. Pierce?
I think Christine introduced that or answered that question effectively. I'm going to echo it. You know, I think, well, I'm certainly motivated by unmet need of the patients that we see, and I think that's true of many physicians. So the vision loss that the patients that we care for drives us to understand the causes of that vision loss, and then hopefully use that information to develop therapies to prevent further vision loss or restore vision. And it's kind of a dream come true to be in a field where that's actually happening, right? As Christine said, we're actually achieving translational research with genetic medicines. So it's a very exciting field to be part of, because I think there's a lot of potential to develop and deliver therapies to our patients.
Thank you. We actually have our first question in a couple of parts, so bear with me as I read this, and I'll repeat it as necessary. In the most recent presentation for ARVO 2020, Dr. Stephen Russell discussed results of a phase 1b/2 trial of Sepfarsin, an intravitreal RNA therapy for LCA-10. According to the results, cataracts were the primary safety adverse event. Is it possible to avoid cataracts formation with the use of lower doses, or perhaps adjustment of formulation? based on other ophthalmic drugs in the use for more common eye diseases? Is it possible to improve it with different drug administration procedure? And I'll stop there to let Steve wants to weigh in on that part. There's a second part to that question as well.
Christine, do you want to start?
Sure, that is a great opening question, whoever asked that question. So starting it off challenging. So let's kind of put it in perspective. So, the types of therapies that we have right now for delivering a gene or potentially modifying RNA include ocular gene therapy, the standard replacement gene therapy, where we do a vitrectomy and then we do a subretinal injection. And then of course we have a risk of creating a cataract with that type of therapy. And I know your question didn't ask that, but I think it's important to point out some of the alternative modalities for therapy right now as far as gene delivery. So that can cause, having had a vitrectomy and a subretinal injection can certainly cause a cataract in a small percentage of patients, and they may need cataract surgery. As compared to what you're asking about with the sepofarsen trials is, so these are intravitreal injections, theoretically office-based, where we can do an intravitreal injection and repeat therapy. A lot of these trials were repeat dosing every 3 months. So either the mode of treatment, so being the intravitreal injection, which is also at the pars plana, which is the part of the eye that's a safe place to put a needle. I do 30+ injections a day when I'm in my busy VR retinal clinic. You're not seeing inherited disease patients. I'm doing these for diabetes and macular degeneration. So it's a very safe injection procedure to do, but in some percent of patients in some trials, they have seen cataracts. Now, is it due to the dose? Probably, in my opinion, it certainly could be dose related. We're not seeing, I doubt the procedure itself of an intravitreal injection, from all the data we have with macular degeneration, is causing the cataract just from the injection procedure itself. However, when we inject saline or BSS, you can look and compare the rate of cataract formation. But it is, from these trials, I think that we learned that dose, and that's why these are dose escalation trials, to try to decide the correct dose. Dose is important. And it's not just a cataract, so there are precipitates seen behind the lens in some of these cases. And then the question of a mild amount of cystoid macular edema or swelling in the retina was seen in some small percent of patients, which was modifiable with giving a small, very quick course of topical steroids, which I give all the time to my patients for various reasons, and really no issue at all with giving a short course of topical steroid drops to these patients to help the macular edema that we're seeing in some percent of patients. But the cataract, the precipitates or the deposits behind the lens were thought to be and seen to be dose related, and Dr. Pierce can certainly chime in on that as well. I'm certain that as we refine the correct dose and find the safe and effective dose, that that can be improved. At the worst, the question is, how bad is it to have cataract surgery? And I think as a vitreoretinal surgeon, we often downplay that maybe more than we should. And we say, it's not that big of a deal. I give patients cataracts all the time by giving them steroid injections or by having to do a vitrectomy for retinal detachment. And then they go get their cataract surgery. And that's a very standard procedure. It's done quickly and safely and done well. The reason it's not, I think we shouldn't downplay it too much though, is it's a bit different for a 60-year-old to have a cataract surgery versus a 15-year-old or a 20-year-old. If you have to have a cataract surgery at that age, we've taken away a natural ability of the lens to accommodate. And so those patients may need to wear reading glasses. If we only do cataract surgery in one eye and the other eye is unaffected, then again, we have the issue of balance and one eye not being able to accommodate, which means hold a paper up to you and be able to read and focus. That's the ability of our natural lens that we have that can do that. We have some fancy intraocular lenses that can sort of try to do that, but they're not quite as good as the natural lens at accommodating. But yes, I think it's something that we should look at and has been looked at. The numbers were very low, and I can't quote you the numbers, how low the numbers were. Maybe Dr. Pierce can. We can look that up, but they were low, and then they were dose related. So I do think that that's something that needs to be and is being considered as we go into Illuminate, which is the phase 2/3, which is now going on. That trial that you recently mentioned was published 2 years ago now. So 2018, we have very good data on the positive on that publication. Multiple patients actually improved in visual acuity. There was one profound responder, one patient who I believe was light perception, who improved to better than 2400 in their visual acuity. So we had an excellent responder in that trial. We weren't even expecting to see that. So there's certainly very, that paper was actually written by Sam Jacobson's group in ArtsDCM, which is a very conservative, excellent group of researchers that never overplay results. You know, they're really a safety group. And so when you hear a positive result out of Jacobson's group, I really take that seriously. He's an excellent researcher, and you know, reading that paper, and again, Steve Russell is the senior author because he was involved. It was through Iowa and Penn, but that paper overall was very encouraging as far as the efficacy data from the phase 1/2 trial. And now, like I said, they are in Illuminate, which is the phase 2/3 trial, to compare 3 different arms. There's going to be 2 different doses and then a control sham arm. But I'll stop and let Dr. Pierce answer as well.
You know, I don't think I need to add to that because I want to make sure there's time for other questions. I think we've covered that topic pretty thoroughly. So Chad, do you want to move on to the next question?
Sure, yes, definitely. The next question is following on to that, how could this adverse safety event affect long-term benefit to risk for pediatric patient populations? Would it be possible to draw any predictions from the existing 12-month data?
So again, I think Christine already really answered that question.
Yeah, okay.
To the
Just want to
issues
make sure we
cataracts
fully
are driven
cover that.
for pediatric populations.
All right, great. The next question is, do you all have any information on the gene RPGRIP1?
Yep. So inherited retinal degeneration caused by mutations in the RPGRIP1 gene have been studied at our center at Mass Eye and Ear for quite a long time. The gene was originally discovered by an investigator, Tiansen Li, at Mass Eye and Ear, and he and his colleagues started developing gene therapy for disease caused by that gene. And we've carried on those studies. There's beautiful proof of concept that preclinical proof of concept that AAV-mediated gene therapy for RPGRIP1 is effective in animal models, and that therapy has been licensed from Mass Eye and Ear through Odilia Therapeutics to another company to develop clinically. So PTC Therapeutics is now working on developing AAV gene therapy for RPGRIP1 associated disease and hopes to have clinical trials going in the next 2 to 3 years.
All right, thank you. The next question is, hello, doctor. My 3-year-old son diagnosed with LCA. He's genetically tested 2 years ago, and results came back with 2 variations, the Nmnat1 mutation and 1 indeterminate. Is there any research in progress for the Nmnat1 gene? If yes, kindly let us know the current situation. What tests need to be done to know the exact mutation?
So Christine, since we're working on Nmnat1, why don't I start with the gene therapy? But it'd be great if you could maybe answer the second part after I discuss the Nmnat1, because I think that's an important topic.
Mm-hmm.
Does that sound reasonable?
Yeah, sounds good.
So we and a bunch of other teams found that mutations in the Nmnat1 gene cause severe early onset retinal degeneration about 10 years ago. We developed an animal model of the disease by adding one of the mutations we found in patients to the genome of mice. Those mice get exactly the same disease as people do, severe early onset retinal degeneration. And we've spent the past several years, again, working on AAV-mediated gene augmentation therapy for Nmnat1 associated disease. And I'm very pleased to say that the publication, the paper describing our results and final success at identifying a type of AAV gene therapy for Nmnat1 disease was just accepted for publication on Thursday. So the answer is Nmnat1 gene therapy works, and we hope also to work towards bringing that to clinical trials. The second part of the question here is really important about genetics. So Christine, why don't you take over from here?
Yep. So this is quite common in a very frustrating scenario as a physician trying to counsel a patient through results such as this, and it's actually not uncommon. So to have so we know for these recessive conditions that we're looking for 2 mutations in the gene. Unfortunately, genetic testing is not perfect, and sometimes that second mutation can be hidden in an intron. We can simply not be able to find it with sequencing. So it's not uncommon that a second mutation can't be found, or that a second mutation is either benign or unrelated or in another gene that wouldn't necessarily make sense or explain the condition. So the answer is that likely, first of all, one of my questions for the family would be, and these are kind of conversations, these are questions you can just sort of think about. I don't know if we can go back and forth, but where was the testing done? You know, was it a CLIA certified lab? Was it a comprehensive panel? And, you know, what exact genes were tested in the LCA panel? That's important. Some labs are better than others. You could consider having an ocular genetic counselor. There's a wonderful group called Inform DNA, Carmen Trupek, who's not on this call right now, but she's very involved in Visions and Foundation Fighting Blindness. Might actually be able to help you look at the results. You can actually officially have a referral for an Inform DNA, which is a telecounseling service that offers ocular genetic counseling to help you look at these results and understand them. I tell most of my patients, especially if they have LCA, and we really have a high likelihood of being able to find that second mutation at some point, to have retesting done, you know, at least every 2 years. I don't know if Dr. Pierce has kind of a different model that he recommends, but testing actually improves substantially kind of every couple years. We're on a steep incline in our understanding of ocular genetic disease. So it might be worthwhile to have retesting done at some point in the future. Again, I would recommend that you talk to an ocular genetic counselor or a retinal geneticist well-versed in this to be able to kind of look through that lab result with you. Because, you know, Dr. Pierce was mentioning, which is very exciting, Nmnat therapy, if and when this does exist and is prime time ready for clinical therapy, would be a requirement for, you know, an inclusion criteria for any ocular gene therapy trial to have 2 mutations proven in the given gene. In order to replace that gene, we want to know for sure that the disease condition is related to that genetic mutation. So,
I certainly agree with that. I'm just going to add one piece because we're doing a lot of research on this as well. So we found that in about 1/3 of patients with inherited retinal degenerations, that a non-coding mutation or structural variant in the genome, and I'll come back to that to explain what those are in a minute, is part of the disease causality, right? So genes are made up of parts that encode proteins called exons and parts that don't called introns. And normally when we do genetic testing, we only sequence the exons, because those are the parts that get turned into proteins, which are used for vision in our retinas or function in any other cell type. You can identify the genetic cause of disease about 2/3 of the time if you focus just on those exons, the coding portions of the genes. So a big question in the field has been, where's the other 1/3 of genetic causality? And it turns out that it's pretty likely to be in the non-coding portions of the same genes already associated with retinal degeneration, like Nmnat, but it's in the introns of the genes, or it's because part of the gene is missing. So we and many other people in the field of genetics have been working on methods to identify those so-called hidden or non-coding mutations. And we're now shifting to doing whole genome sequencing for many families that have this sort of indeterminate genetic answer. So as Christine said, I think genetic counseling is absolutely the first step. And then once you understand the situation, if there's a missing mutation or a second mutation that's not found, working towards either sequencing the entire gene for Nmnat or doing whole genome sequencing is likely to be the way this kind of thing is going to go.
All right, thank you both for those in-depth answers. The next question we have is, what's the therapeutic outlook for CRX-caused LCA or LCA7?
Sure. So mutations in CRX are in the kind of unusual category for LCA. It's one of the few dominant forms of LCA, meaning just one mutant copy of the gene would cause disease, unlike recessive disease where 2 mutant copies are needed. So it's likely that the mutation in one copy of CRX causes the function of CRX to change. Instead of helping vision, it hurts vision. So we probably have to remove that mutant copy. And this is where I think gene editing with CRISPR-Cas9 mediated approaches, like one of the trials for CEP290 associated disease, is going to be needed. Fortunately, CRISPR-Cas9 mediated gene editing or prime editing is exploding in terms of research. It's already being studied for CRX-associated disease, and I'm optimistic that it will also be used clinically to treat disorders like CRX-associated retinal disease. Christine, anything you want to?
Just from a genetic testing standpoint, of course, again, the family counseling is very important for this as opposed to other types of LCA to confirm the family history and the pattern, to understand 50% risk to offspring and future offspring. So just make sure that you're aware of the fact that it is inherited in a dominant fashion and have had appropriate ocular genetic counseling to understand risk to future children or other family members who may be affected and need to be tested.
All right. Thank you. Thank you both for that. And Dr. Pierce, since you mentioned sort of CRISPR-Cas9, it sort of sets us up for the next question, which is essentially talking about emerging technologies or emerging therapies in this space, and then sort of the approach how we take them. So this individual would like to know, is the field going toward addressing IRDs one gene at a time, or are therapies that will treat independent of disease phenotype or disease cause?
I mean, it's a great question. I think the field is going to continue to do multiple things at once. I wouldn't say that we're really transitioning away from augmentation replacement gene therapy. That's still sort of the standard of care for gene therapy at this point with the, previous FDA approved therapy. We still have multiple replacements. So when we use the word replacement, we mean for augmentation gene therapy to replace the full gene for a recessive disease. So that's still the most common. We still will do that. The FDA, understands our pathway forward, and Wiley Chambers understands the pathway forward to have FDA approval for replacement augmentation gene therapy. However, in addition to that, and I wouldn't say to replace that, but in addition to that, we're learning that there are multiple other helpful potential modalities that aren't as well established, but they're certainly coming along quite quickly. So the therapies there would be the CRISPR-Cas9 gene editing. And the idea there is to potentially be able to address dominant disease. There's of course antisense oligonucleotide therapy, which we should discuss today, due to the CEP290 trials. That was our first question. So that's antisense oligonucleotide, being able to address at the level of RNA and transcript modification rather than to long-term expression change the level of DNA. And then, there's a whole other field of optogenetics. So for patients who have lost significant function and have no remaining photoreceptors or RPE cells, being able to turn the remaining healthy cells that are still present in the retina, for instance, the ganglion cells, on as far as being able to have some phototransduction capacity with channel rhodopsin. So those are multiple different trials that are not genotype specific, really only require lack of function, and remaining ganglion cells or potentially other bipolar cells could be targeted to turn on a phototransduction mechanism. And that would be really very early work being done in optogenetics. The study there being right now on retinitis pigmentosa with early work, and a lot of that out of Dave Birch's group. But so I wouldn't say that we're really moving away from replacement, but I just think that we're learning that there are other types of modalities of treatment, and that has really just opened up the horizon for us to be able to treat different types, for instance, dominant disease, and then also non-genotype specific treatments.
I think I'm going to pick up on that answer and add two other categories of potential gene-independent therapy. But I certainly agree with Christine's point that I don't think the field is moving away from gene-specific therapies. Gene-specific therapies are turning out to be effective, and I hope we continue to use them and develop them. But we also need additional therapies for people who don't have a genetic diagnosis, or to complement and be used in combination with gene therapies. So there are gene-independent therapies being studied to preserve sick retinal cells to make them function and stick around longer. They're called neuroprotective therapies. There are two different clinical trials of those. We'd be happy to discuss if there's interest. And then I think I put stem cell therapies in this category as well, right? If we could, instead of fixing sick retinal cells with a genetic therapy in someone's retina, if we could add back new retinal cells to replace ones that are lost, that is thought to have the potential to restore vision. And there have been a couple of clinical trials of that and more to come. So the answer is yes. There are therapeutic modalities that don't depend on knowing the genetic cause of disease, which are being developed, and I think will be deployed successfully long term.
Thank you. Maybe I'll ask a follow-up question as far as this category. On Thursday, we had Dr. Kapil Bharti on talking about the advance of cell therapies. And a dominant question during that session was, well, if this treatment, cell replacement proves out to be, it works great, we're able to do it, would you foresee in the clinic actually having to marry that with gene therapy as well? So what does it look like between the different technologies? Would you have to marry those together if somebody had a particular genetic mutation that's causing cellular degeneration? Would you then also have to fix the other cells around it to prevent it? Could you give us, the audience, a little bit of flavor for how that might work in the future?
So actually, you don't have to use them together, but I think you can, is what I'd say. So one of the exciting aspects of the potential to treat the genetic causes of early onset severe retinal degeneration, which traditionally has been called LCA, is that a lot of patients have intact retinal structure, but poor vision. So that if you fix the underlying genetic cause of disease, you can restore vision and preserve vision. That's what's been observed with RP65 gene therapy. There's evidence that that's starting to happen for RPGR therapy, and we hope that continues for other genetic forms of disease like RP group 1 associated disease, as we discussed earlier. So for some people, the genetic therapy is going to be enough, right? Especially if we treat children before their disease is advanced. So I think I would say you could start with a genetic therapy, see how you do. If that isn't enough, or people want to consider potentially trying to achieve more vision, then you certainly could follow with a stem cell therapy if those are shown to be effective and try to expand the area of preserved vision. And I think those therapies will be used together going forward, but we need to understand how to use the therapies separately first before we can combine them. Christine, any thoughts?
Well, yeah, and I totally agree with that. And I think, of course, later stage disease, like you mentioned, might need combination therapies. As we move things, patients always ask me, why can't we have the FDA trial where we do 2 at once? And the answer is the FDA doesn't work like that. So when we have something that's going through FDA approval process, it really needs to be one indication with all of its preclinical data and pre-toxic testing. And then we're going to move that forward, phase 1, 2, 3 trial process and see if it works. So you can't test 2 different things at once. Even it gets complicated trying to test if you have a novel device or delivery device and a certain therapy. So the FDA really wants one thing tested, and that makes sense. You don't want to have confounding variables if there's a safety issue. So it's really sort of test one thing at a time. But it is true that potentially if multiple things were FDA approved and available combined, then at that point, if things are commercially available, we're a little bit more lax on being able to administer different therapies, judgment as vitreoretinal surgeons and IRD specialists to be able to administer multiple therapies at once, or the thought of adjunct therapies, for instance, and I know that there's been multiple changes with the company with QLT, Novelian, et cetera. I'm actually not even 100% sure where that's at, but the idea of, a patient who's had ocular gene therapy with RP65, they also have adjunct therapy with a QLT type compound, which is an oral visual aid modifying type drug. And the answer is yes. I mean, if that were ever FDA approved and available, you can combine therapies. I also think it matters, what do you mean exactly by stem cell therapy? And I wish I had a horrible day in clinic on Thursday, so I did not get to get on that talk, and I really wish I had missed it. Maybe it's on video recording. I have to watch.
It's next.
Is it? But stem cell therapy, I think you have to sort of understand that stem cell therapy doesn't equal all stem cell therapy. So is it stem cell derived RPE therapy, which are some of the trials that are going on right now in the J-site, Reneuron, or is it actually, is it a retinal progenitor stem cell? Is it a retinal progenitor cell? Is it in the future, and this is sort of happening as well right now, is it a stem cell scaffold where we're actually surgically transplanting a piece of retina? So there's such a huge expansion right now in the field of stem cell therapy, but in order to actually be able to replace the gene at the as well as be able to use stem cell therapy at the same time. You know, those therapies, again, have to be walked their separate way through the FDA process, and it's only really after FDA approval that you can combine the therapies.
Thank you for that. And yeah, well, I think we'll pull that thread a little bit more as far as stem cell treatments. We'll start with Dr. K and then go to Dr. Pierce, but I just want to sort of share our own little personal sort of story and then sort of direct the question to you all. Recently, my mother saw on Facebook that there was amazing stem cell therapy for her knee. So they could come in, they would do something to inject stem cells in there. So she went, they injected her with something, nothing happened. And I told her, and I was like, well, did your physician refer you? She said, no, I saw it on Facebook, it looked great. So it seems that the stem cell field is sort of exploding, but at the same time, I think patients are having a hard time sort of deciphering, you know, what is really the good treatment and the good clinics to go to for these therapies, or are these therapies actually even approved at all? So Dr. K, could you start off and sort of mention that? Because in Thursday's session, it was mentioned that there were no FDA-approved stem cell trials. But are there ongoing trials currently? And how can someone decipher between something that's legit versus something that is just being sort of advertised through social media and whatnot? Yeah.
And I live in an interesting state, to give you some historical perspective and actually relatively current perspective. So I live in Florida. And so if you're aware, there was a company, and I won't really use names right now, but there was a group that was putting forth advertisements for stem cell therapy that involved an either intraocular, retrobulbar, or vitrectomy-related subretinal injection of, quote, stem cells. There were multiple patients who were treated in an office by a PA, not an MD, but a PA who had intravitreal injections of, quote, stem cells. And by that, this particular trial, which somehow made its way unbelievably onto clinicaltrials.gov and had I don't know if it had an NCT number for it. I don't think it had an IND, but the overall point is that you have to be very, very, very careful with stem cell therapy, because long story short, multiple patients had their blood extracted, spun down, and then literally whatever this lab was that was doing this was injecting, quote, stem cells, whatever that they thought that meant, back into the patient's eyes. The patients also paid $20,000 to have this done, which is a major red flag. Any good clinical trial should never ask you to pay money. So if any trial asks you to pay money, steer clear. Also, always ask if it is on clinicaltrials.gov, it has an NCT number, National Clinical Trial Identifier number. If it has an IND, Investigational New Drug Application, through the FDA to approve it being done. More importantly, talk to somebody like Dr. Pierce or myself or an IRD specialist and make sure that we agree with it and recommend it and think it's good for you. Because I didn't luckily have any patients of my own who did this, but I've seen patients now post-operatively. Those patients who had those injections developed within a day a flagrant inflammatory endophthalmitis, and they developed profoundly aggressive retinal detachments. The eye basically collapsed. One of my partners who lived in Miami at the time, actually at Bascom Palmer, tried to repair some of these detachments and was unable. He was, of course, not at all involved in having the patients treated. He was trying to save them after the fact, and they were really unable to repair these aggressive retinal detachments. So multiple patients went bilaterally blind, no light perception blind. Lots of lawsuits going on of that in Florida for that right now. But the point is, there are some very unethical people out there, and there are some very scary things that can be done. You have to be very careful and cognizant as a patient or family to really make sure that what you're entering is an actual clinical trial, and with ethical, upstanding individuals involved, especially in the field of stem cell therapy, because there's a lot that is posted out there that is not medically sound or safe. So really run everything you do. Honestly, it doesn't take much more than an appointment with an IRD specialist to discuss it. Somebody, or even a phone call with someone at Foundation Fighting Blindness, to have somebody fact check what you're going to get yourself into and make sure that it's an upstanding clinical trial before you jump in.
And I guess I'll pick up maybe on one part of that, just to point out that there are FDA approved clinical trials. You have to be careful with how you use the term FDA approved, right? So in order to do a clinical trial, in the United States, you need, as Christine said, approval, you need an IND approval, investigational new drug approval, which means you have to tell the FDA what you're planning to do. You have to tell them that it's safe, and you have to provide some evidence that it might be beneficial. So there are some, and when you do that, you list your trial on clinicaltrials.gov, as Christine mentioned, and get a clinical trial number assigned. So there are clinical trials of stem cell therapies that do have INDs. Just as an example, at Mass Pioneer, we are one of the centers for the Renuron-sponsored study of human retinal progenitor cells, a different type of retinal stem cell, for the treatment of retinitis pigmentosa, and what we would call a rod cone degeneration, a later onset disease than the severe early onset disease that's typically called LCA. That trial, we've done the phase 1/2 part of that trial. We're writing up the results, and their company is planning a phase 2 extension of that study, as far as I understand. So that's an FDA, not approved. That trial is being done with the correct procedures, according to FDA guidelines, to make sure that it is safe and isn't going to cause harm like the kind Christine described. Chad, do you think that addresses the stem cell question?
I do. Can y'all just expand? I think this is such an important topic. I think many people are interested. So I'll just ask the stem topic just a little bit longer and just ask a follow-up of someone entering into a phase 1, maybe if they're applying for a phase 2 or phase 3, what should they expect in each of the different phases? So like if I sign up for a phase 1 clinical trial, should I expect any results, or what is typical experience for a patient participating in a phase 1 trial?
I think the experience varies a lot. The common piece to all these phases of trials is, number one, they're voluntary, right? The most important step, first step in considering one of these trials is informed consent, right? So you have to sit down with a group that's your care provider that's doing the trial, hear all about it, ask any questions that you have, and if you're interested in participating, give your informed consent that you understand the risks and potential benefits of this study. That discussion, when I do it, usually takes an hour to an hour and a half for people in a phase 1/2 trial, because we have long discussions about the potential risks and the potential benefits. And really, I think the decision to enter into one of these trials is an individual risk-benefit calculation. Are the potential risks worth it compared to the potential benefits? And you can't make a global decision about that. That's an individual choice. There are many steps that go past that, but maybe we'll just stop there to say, you got to understand what you're getting into and understand what are the potential risks, what are the potential benefits, does that work for you? Maybe Christine, you could pick it up from there.
Yeah, I'll just add one thing. If you wonder why we often see on clinicaltrials.gov for ocular trials, phase 1, 2, and combined, it's just very common for ocular trials, at least for these IRD rare orphan disease trials, to combine phase 1, phase 2. If you're thinking specifically about a phase 1, often those are actually testing a therapy in control patients and normal patients. So Alkeus with this oral Stargardt disease drug did a phase 1 in patients without Stargardt disease first, just to look at doses. But that's rare. Most of our, for instance, treatment ocular gene therapy trials are going to be phase 1, 2 combined. Still, so what that means, phase 1, 2 is usually the first time it's in the human eye. It's been tested in mouse model, large animal, toxicology tested in non-human primate. And so then we submit an IND to the FDA. They say, okay, go ahead with your trial. And then we test first time in the human eye. So again, long discussion. I have these consents discussions with my families as well. Typical to Eric Pierce, I probably spent about an hour with these families talking about consent, but they're going to basically, phase 1 and phase 2, we're going to look at primarily safety will be our outcome measure of our primary outcome measure. And then there'll be secondary outcome measures like visual acuity, microperimetry, other things. But primarily we're looking at safety. So risk benefit discussion there is very important just to understand the potential risk, because it's really still the first time in the human eye. Now there might be previous patients, you might be patient number 14. And so I usually have relatively heart-to-heart conversations with the patients, and as much data as I'm allowed to give them within a trial. I give them all the information of the previous 13 patients treated in the trial and how they've done. And so that's really confidential data that you're privy to once you're sitting there in the room with the investigator, signed an informed consent and discussing whether their child should enter a trial. When you're in a phase 3, you have a lot more information, because to get to a phase 3, you've had the FDA look at our phase 1-2 data, decide to go forward and allow the company or sponsor to move into a phase 3. So that basically, and it's gonna be a larger number of sites, typically larger number of patients treated. Typically they're gonna be kind of going for FDA approval at this point. Whole long discussion of how many, sometimes it takes 2 pivotal trials, sometimes it takes 1 for the FDA to approve rare orphan disease, but a phase 3 in general, the primary outcome measure is now suddenly gonna be efficacy. And so we're gonna be looking with enough number of patients, like 45 or so, typically for an ocular gene therapy, to be able to move to pitch this to the FDA as, you know, is it statistically effective, and are we seeing a statistical benefit? And so efficacy is the primary outcome measure. You're still looking at safety, of course, and all that data has to be submitted to the FDA advisory committee, you know, prior to FDA approval. So that's really the overview there.
All right, thank you. And thank you both for spending such a great amount of time on that. But I feel like it's so important for our constituents to know a lot about the process. So thank you again. We're gonna switch gears a little bit. One of the attendees has 2 sons affected by a CEP290 mutation. Is their understanding that the gene editing technology being developed are geared toward addressing a common mutation in CEP290, and wants to know, well, what about patients who have other mutations in CEP290? And is there any hope for that type of treatment being applied to them? And they put it out there that, would it be possible sort of to take their stem cells and somehow develop an eye cup or and test drugs or things from that pathway on that particular genetic mutation that is affecting her sons?
So I think we got it. They're both great questions. Let's take them separately. So the first question, just about what about therapies for other mutations in CEP290? The answer is yes. There's definitely hope for treating those. It's very well understood in the field that the antisense oligonucleotide therapy that Christine discussed earlier, or the Allergan Editas-sponsored clinical trial of gene editing for CEP290 are, as you said, focused on a specific common mutation in intron 26. But that's really meant to be a proof of concept. And both, actually I would say both are meant to be proof of concepts. Those therapies work for that specific mutation. I think they'll very quickly be applied to other mutations, both in the CEP290 gene and in other genes. So if gene editing works for the common intron 26 mutation in CEP290, it could be applied very quickly to other types of mutations in the CEP290 gene, for example. But in addition to that, there are other investigators working on other types of genetic therapies for CEP290 disease specifically. I'm currently on the retinal disease study section for NIH, and I think it's okay for me to say I've reviewed several grants in the past year looking at other approaches to treat other mutations in CEP290, some of which look very promising and I believe have been or are going to be funded. One of those, for example, would be taking the CEP290 gene, which is too big to fit into a single AAV, and breaking it up into 2 AAVs, giving those both to the retina and allowing the gene to kind of recombine in retinal cells. That approach has been used for other genes like ABCA4 and will be studied clinically for that. That's also being applied to CEP290. So the answer is yes, therapies for other mutations of CEP290 are being developed. The second question, what about iPS-derived retinal organoids? Christine, do you want to take that or should I keep going?
No, go ahead. I'm going to add something, but go ahead.
There's a lot of interest in the second topic asked in this question. Can you take someone's stem cells, make little eyes in a dish, retinal organoids, and test therapies in them? The answer is yes. And that's being done in general for multiple different diseases and is also being done for CEP290.
And so, yeah, just one follow-up on that. Ed Stone's, again, having did my fellowship at University of Iowa, probably one of the most active groups, in addition to Dr. Pierce's group, for IPS and trying to study a certain genotype in a dish is Ed Stone's group at University of Iowa, the Carver lab there, very actively involved research group. So the idea there, when a patient goes to Ed Stone's group at University of Iowa, they, and as I was a fellow there, we draw their blood. And so we're actually taking a blood sample, testing for genetic defect, but also spinning down and identifying taking a skin sample. So take a skin sample to identify, so a little chunk of fibroblasts, and being able to basically identify and induce pluripotent stem cell from that, and then potentially replicate or recapitulate the disease that the patient has, because that would have the same genetic defect within that cell, sort of the idea of testing a genotype within a Petri dish. And so, Ed Stone is really one of my mentors and great leaders in this field, who's really probably one of the predominant leaders in that iPS derived gene testing idea of being able to look at a particular genotype within a dish from a patient's skin cell. So very active field, really a little outside of what I do as a clinician here, but very active and exciting field. And I did want to just point out one thing, which is that I was also discouraged when I said, oh geez, all these trials are not only genotype specific, but mutation specific. But with that said, at least for CEP290, that is the one that we call a founder effect. So that particular mutation is extremely common. Maybe 15 to 20% plus of patients have that particular the intron 26, the C2991 mutation. So it is very common. So it certainly made sense for the companies to pick that. And it does have this splicing defect, which basically, if we can alter the splicing defect, we can prevent this abnormal frame shift stop codon from occurring, and be able to modify and allow the gene transcript to be to create protein. So that was a good selection. As far as, it's a very common disease, but it's encouraging. And I also had heard the same thing, but the idea that this is proof of concept, and these companies, not just ProQR and Editas, but other companies are very interested in the concept of being able to move this field forward and pick more and more mutations. And of course, we're not talking about USH2A today, but the same thing with USH2A, being able to pick a specific mutation. But there are certainly other mutations within these genes that would be amenable to the same type of antisense oligonucleotide therapy.
All right, thank you. This question actually speaks back to the Nmnat1 mutation. And the question is, this attendee's son has been diagnosed with this mutation and has been told that the retina is not fully developed. So the question is, if there is a therapy for this mutation, is there a chance that his son can regain vision?
Right. So first, I see in the question answer that this particular attendee has been trying to get in touch with us. I apologize that we apparently didn't respond. We do get many, many inquiries. We try to respond to them the best we can. Sometimes we miss some, and I apologize. We'd be happy to try to respond going forward. But I agree, I'm glad we're connecting via this meeting. So you are correct that Nmnat1 associated retinal degeneration is one of the types of severe early onset disease that can be associated with what's called macular atrophy. What that means is the center of the retina, which is called the macula, where we have our clearest central vision, can become atrophic, meaning it can become thin. The retinal layers, instead of being the normal thickness, can be thinned almost to a scar in some cases. For Nmnat1, we don't believe that that's completely developmental. It seems to be acquired. So one option would be find patients when they're children before they get this atrophy and treat them and hopefully prevent that. We do see this present early on in some patients, both with Nmnat1 associated disease and some other forms of severe early onset disease. For example, some patients with CRB1 associated disease have the same type of macular atrophy. So our thought about treating those patients is, we know we're not going to be able to treat the macula because the retinal cells are already too damaged. But when we've looked, we have seen intact retinal layers outside the macula in the more peripheral retina. And our thought would be we could apply the gene therapy there to restore some vision, which we hope would be beneficial, even if it's not central vision.
All right, great, thank you. And I think there's a follow-up question to this particular gene. And where is the best place that we are able to access regular research updates to understand what's the most sort of emerging data and therapies that are coming out?
So to me, this is where relationships with inherited retinal degeneration providers are most useful, right? So when our patients ask me that question, I say the way you get these kinds of updates is during your follow-up visits. When you come to see us every couple of years, we can have these discussions about what's going on with research. Beyond that, there isn't actually the best forum to get general updates about inherited retinal disease research is the Foundation Fighting Blindness. The team at FFB does a great job maintaining their website. The team of science writers thoughtfully interprets the literature and news and provides updates. That's a great source. Beyond that, you can reach out to individual investigators, but as apparently was the case this time, it doesn't always work because we can't respond to every email that we receive as much as we try.
Yeah, I agree with that. I would just echo the Foundation Fighting Blindness. Not just the website's excellent. I mean, I actually go to the website myself to make sure I haven't missed an update. So I mean, it's a very very good website, very user friendly, and often has excellent papers that you can click through to. I would also say do this with caution, but you can also, if you found out about a certain trial, then you could go to the sponsor's website, because often there will be, now do this with caution, because again, you're going to directly to a sponsor website, but the sponsor websites will often have their preclinical data, they'll have their pitch decks, whatever is publicly available information. So for instance, we're talking about ProQR today. You could click on the ProQR website and see what clinical trials are going on. You can even see what's in the pipeline. So sometimes that, and again, I think you should do that with caution, because I think the number one point that Dr. Pierce is making is you want to go through your IRD specialist kind of for advice and to see what we recommend. Also, in addition to the FFB website, as you all probably are aware, the Visions Conference is once every other year, but then there are also community-based, for instance, I live in Gainesville, we have a really active group in Jacksonville, a really active group in Tampa as far as the FFB, and so they often invite me and I drive over there and give a talk, and so every few months, or it's less often now with COVID, but we used to go and give talks in person at Visions, at FFB sponsored events, at the different FFB chapters within the communities and and big cities. So definitely look through the FFB website and find your nearest chapter. That's a great way to get involved. And the FFB is, a very ethical group and can give you kind of a good broad overview of what trials are going on.
And yes, and I'll just put something, FFB being a great resource, as we definitely try to champion our community and make sure that you get all the information that you need in order to, feel satisfied with understanding whatever sort of disease phenotype that lands under the IRD space. And if we don't have the answer, we will work very hard to find the answer for you, because we have such great networks with the academic and sort of drug industry that are working on solving these problems. The next question is, what is the best way to contact Mass Pioneer to learn more about the work they're doing in RPGRIP1? And this person is interested because they've been recently genetically diagnosed with LCA.
So again, we're happy to try to respond to inquiries. I actually have a session every week where I sit down with our genetic counselor and we respond to all the messages we've gotten in the past week. And we often spend a considerable amount of time doing that. We don't get to them all, apparently. I apologize. So we're happy to try to respond to emails. I'm not sure in the RPGRIP1 case that we're the best source. As I've said, we've now licensed this to a drug company that's developing it for clinical use. So I think that getting updates about RPGRIP1 will shift to looking at the biotech pharma companies that are now working on that, Odilia Therapeutics and PTC Therapeutics.
All right, thank you. The next question is, if you participate in a phase 1/2 trial, are you automatically enrolled into the phase 3? And then what about the placebo group? Is it possible that you would be placed in that group and not in the actual treatment group?
Okay, I can speak to this one because, so I'm an investigator for multiple trials right now. We're doing the, for instance, the Biogen, which is a phase 1/2 RPGR study, which is now completely enrolled and will be moving into the phase 3. So for instance, for the Biogen trial, the phase 1/2 had a placebo. Well, it had we called it a control arm because it wasn't that we were doing any sham injections. So we had patients that were randomized 1:1:1. So there were 3 arms. So there was dose 1, dose 2, and then there was a control arm. And so unfortunately, patients, and you have to explain this to patients, and that's part of the informed consent, have a 1 out of 3 chance in that particular trial of being just watched as a control. In that particular trial, there was no guarantee that the patient would then be treated in a phase 3. So I'd say the short answer is no, there's no guarantee that if you've been in a phase 1/2 that you'd be in a phase 3. In fact, it's sort of the opposite. If you've been treated in a phase 1/2, particularly if you've had a unilateral treatment, you might very well not be able to be enrolled in either that phase 3 or in another company's phase 3, because it often is an exclusion criteria that if you've had any prior therapy, particularly any permanent surgical therapy, if you've had an oral drug, we often allow a washout period and you can get into another trial. But if you've had a vitrectomy and a subretinal injection of a gene therapy, it would be very difficult and confounding if we were to try to put you in a second trial or even in a phase 3 trial. Now, a lot of companies do what we call follow-on studies, so we still can keep you involved and treat. And even in the follow-on studies that Spark did, they would treat the second eye of the patient. That doesn't happen with all companies, but it happened with Spark that the second eye was treated in a follow-on study. Was treated a year later after the first. That was a Jean Bennett paper, and one of the important publications that helped pave the way to doing their phase 3, allowing bilateral treatment 1 week apart. But you know, and most of the ocular gene therapy trials that are doing phase 3 are offering bilateral treatment with some separation between time of each eye having the treatment. But even those trials are typically randomized control. Oh, did I just mute myself? Am I muted?
You're good now.
Was I muted for a long time?
No, just for a moment.
So all of the trials that I'm involved with, and as we look at protocols moving into phase 3s for gene therapy, will allow, so what Spark did, which is really an excellent model moving forward, is have the treatment arm treated at 0.0 and have that population of patients who are randomized to control wait 1 year. And then at 1 year, we call that the intention to treat arm. So they're able to have treatment. So the control arm at 1 year is able to have treatment and even have both eyes done. And Spark's situation was 1 to 2 weeks apart. Lots of other companies that are moving into phase 3s right now are likely going to do a similar model. And the FDA, again, once we move something through the FDA and the FDA likes something, other companies tend to follow suit with their model for moving a product through into a phase 3. So I wouldn't be surprised if some of the RPGR studies are going to do similar models where even if you randomize control, so patients don't receive therapy in the control arm up front, maybe 1 year later, they would then flip into the treatment arm. So that's actually quite common for phase 3s. But those are all very important things, you know, back to that informed consent discussion and really carefully making sure you understand all of these things and you have all these questions answered. And one thing I always tell patients when I'm getting them into a phase 1/2 is I make sure they understand that they may not be a candidate for future trials. They could always get a commercially available drug because nobody's going to ask if they've been in a clinical trial, but they will definitely ask if you've been in a previous clinical trial, if you're going to be trying to get into a future clinical trial, and you may be excluded. And so that's very important to understand up front and to have that consent discussion with your investigator.
All right, thank you for that answer. The next question is, our daughter was diagnosed with CRB1 at 8 months. She is 1 year old now. We know there was an approval for a clinical trial in the Netherlands in 2023. How long does the entire process take from the beginning of a clinical trial to the end? And if it shows success and it moves to the US, is there a minimal age to participate? And I'm assuming that you're talking about in this particular mutation.
Right. So I'm going to start with a sort of a little bit of a preamble to say there is work on potential gene therapy for CRB1 associated disease in the Netherlands. There's also work being done by other groups, including in the US, So I hope there'll be actually a couple of different potential genetic therapies for CRB1-associated disease coming forward. So I understand correctly, the question is really asking how long does the clinical trial process take from phase 1 through 2-3 to approval? And the answer is, I don't think we really know for sure. It took about 10 years for RPE65-associated disease. We're hoping we can make that a lot shorter, given the positive experience from that and other trials. Maybe 5 years, but I think it's going to depend on the trials and individual circumstance for each gene. Is there a minimum age to participate? Usually not, right? Most of these studies, well, I should say that yes. Most of these studies are focused on genes that, in which mutations cause severe early onset disease, do include children, but, and Christina, I look forward to hearing your opinion about this. Most of the retinal surgeons that I speak with say they would prefer to wait until kids are a couple of years old to do a vitrectomy and a subretinal injection. So I think most of the trials do have some age limit, but Christine, you're the vitreoretinal surgeon, you probably know better.
Yeah, I mean, I certainly would prefer, I mean, I think it's definitely not advisable to operate on anybody younger than 1. If you look at the SPARK trial, and even, 2 and 3 is still a bit young to operate on the eye. The eye is still, developing up until age 1. So, but I think that 3 and above would be relatively not easy, but certainly able to be done as far as a vitrectomy and a subretinal injection on a 3-year-old. The SPARK trial did allow 3-year-olds to be enrolled. With that said, the youngest patient treated in the phase 3 was a 4-year-old, and that was really more to do with the fact that they needed the patient to be able to do their primary outcome measure, which was a multi-luminance mobility test, and to be able to follow directions. And it was extremely challenging to have 3-year-olds be able to follow directions. I'm a mom of young children. I can't imagine my 4-year-old even doing that. But so part of that was the phase 3. But it's from a retinal standpoint, from a surgical standpoint, I think anybody 3 and up could have a relatively safe vitrectomy surgery, but I would definitely not operate on a 1-year-old or younger. And plus the retina is still developing. So we're kind of, you're treating cells that are still in development at that age. And so there's potential decreased dose and efficacy if you still are in a developmental retinal state.
And just to, I think, pull on that thread a little bit about sort of genetic therapies, can you give me a sort of a feeling for how you feel like this will progress? Like how early on with the advance of technology do you think that we could start seeing interventions as far as like during gestation or, when? Like is there any sort of, and this is more of a futuristic view of it, would you say, about anything on the sort of past the horizon ability to do the gene editing? Or are we just at that moment where we're just starting to understand it and make sure that it's safe? So sort of looking that far out might be a little nonsensical at this time.
I think Christine gave a great answer about the safety of subretinal surgery in children. And I think her answer sounds very clear that really prefer to do that in kids 3 and up. So I think that's one limitation. If we have other modes of delivering genetic therapies, you could consider treating earlier. Your point about gene editing is actually really important, right? So think about this. The retinal disease field is leading the way, or helping lead the way, in developing genetic therapies, right? Luxturna is the first approved gene therapy in the United States. The Sub-290 trial of gene editing that Allergan is sponsored by Allergan and Editas is the first trial of using in vivo CRISPR-Cas9 gene editing ever in people, right? So the subject treated in Oregon was the first person, not just in the United States, in the world, to get CRISPR-Cas9 gene editing in vivo, right? We're thrilled to be participating in that trial. We're looking forward to seeing if the therapy works, but I hope that makes it clear that we're just getting started with gene editing for these kinds of diseases. So we hope it works well. We hope it can be applied broadly, but we have a long way to go to know if we could treat younger and younger kids, or if there's a way to eventually transition us to treating children younger than 3.
And I think
does that address the question you were asking? Sorry, go ahead, Christine.
Yes, it did. Please go ahead, Dr. K.
I completely agree. I would be hesitant to do any gestational treatment, you know, for multiple reasons, and I can't imagine the FDA being too amenable to that idea. But I mean, it's certainly at some point in the future, you can certainly imagine something like that, especially for early developmental problems, even CRB1, when some of the developments and other types of vitreal retinopathies where we see very early malformations and congenital malformations. It would be nice to be able to treat as early as we could, but I think that's a ways off in the future. Yeah, so.
Okay, so right now we have one question left from the audience, and it's a follow-on to the Nmnat1 treatment, and they would like to know an estimated time frame before you would see an FDA approval, and I'm assuming of an actual therapy.
Sure. So as I described, and I will either via the question and answer here or separately try to follow up with this participant who's asked such great questions about Nmnat1 disease. In terms of when is this going to be available for clinical use? So as I said earlier, we just figured out after 3 years of work how to get an Nmnat1 gene therapy to work in a preclinical setting. And what preclinical means is in the lab in Nmnat1 mice. So we now have to say, okay, is the therapy we found that works in mice ready to try to use in people? And if it is, how do we get it to people? We've learned that that's not what we're good at in an academic lab. So that's why we're so excited that companies like the companies we've been talking about, Spark, HTC, Allergan, Editas, ProQR, many others, a whole series of biotech and pharma companies are interested in helping develop therapies in this field. So if, sort of in an ideal world, we would eventually find a commercial partner for an Nmnat1, either independently or via FFB's venture capital approach. And then they would have to do the preclinical work to support an IND and get to a clinical trial. So I think it'll be several years before we're in a clinical trial. I know that sounds like a long time, but at least I think it will happen now. It's not a question of if, but a question of when. And then, you know, as we've already talked about, the whole trial process takes a number of years before there's approval.
Okay. We have another question here. I think it's going back to, and this is actually in the sessions chat box, not the Q&A box, but this individual wants to really truly understand for RP-GRIP1 therapy, would that also be sort of therapy that would be available for children as within the age of like 3 years old? So could you sort of speak to that as like, in general, you know, if you're looking at these types of like gene therapies or different therapies, what is sort of the limits on age, if you could sort of speak to it from that realm?
So I can take a stab at that. So first of all, let's just talk about what we mean by therapy. So if we're talking about the initial phase 1/2 trial, which will be the first time this is potentially available to be used in the human eye, we always start with adults. So there'll be typically phase 1/2 ocular gene therapy trials will be dose escalation trials where we treat above 18 for the first several cohorts, maybe even 3 different dose cohorts of a small, medium, and a large dose. At which point the last cohort, and we actually have to go to the FDA and communicate with them prior to even going into the pediatric dose. We picked our max tolerated dose, and then we move into a pediatric cohort, and then we start with less than 18. So most trials, I would say 6 to 18 is a common age range for a pediatric cohort, which is again at the very end of a phase 1/2. It's not until we start talking about age 3 or 4, we're talking about the phase 3 trials typically. The last thing we want to do is go put a new therapy, a new AAV therapy straight out of non-human primate testing into a 3-year-old in a phase 1/2 for obvious reasons. We're really interested in safety. So it's really usually not going to be until a phase 3 that we're treating. We've already figured out our correct dose, and then phase 3s often will just have one blanket enrollment criteria. Again, Spark was age 3 and up, but their youngest treated was 4. So just recall that we're talking about a phase 3 trial there. So that's multiple years of treatment trial that occurred prior to that were phase 1, 2. And then when something gets FDA approved, when it's actually, quote, available, or commercialized, then the FDA will use that phase 3. They'll often, and insurance companies will use it as well, they'll use the inclusion criteria from the phase 3 to cover the therapy. So for instance, now this was interesting, I was involved in the FDA advisory committee with Luxturna. They were actually, the FDA was actually debating having an upper age limit and saying that we're not going to allow therapy if you're above a certain age. And we all, as IRD specialists, did not want to have that happen because we wanted it to be available for all patients. But they did come up with the statement that you have to have remaining viable cells to have treatment, which makes sense. So there's no upper age limit. And even though there's no, quote, lower age limit on the FDA label, there's a recommendation to basically follow the inclusion criteria of the trial for safety reasons. And then insurance companies can actually say, well, you know what, the youngest patient treated was actually 4, or the inclusion criteria said 3. And so insurance coverage can actually end up covering just a particular age range based off of the inclusion criteria. So all these things are what companies are thinking about as they create their phase 3 inclusion-exclusion criteria of what age it's going to be available for. So good question.
Thank you for that. And this question is, I think, a very good one and be directed to both of you. And it's, as caregivers, what can we do to move therapies from the bench to the bedside? It is very frustrating to know that there are AAVs, mouse and canine models, but there's very minimal movement forward.
Actually, there's a lot of movement forward. I know it's frustrating. I mean, when I sit with patients in the office, I did this yesterday, and someone says, when is this therapy going to be available? What I want to say is, let's do it today, right? I wish we could do that, but we can't quite yet. I think eventually we will. But so I know it's frustrating, but I hope it helps to know that it's happening, right? So again, it's not this question of will there be a therapy? It's a question of when there'll be a therapy. It's really interesting listening to the questions, hearing the questions today and having these discussions, because several years ago at sessions like this at FFB, we wouldn't be discussing when is this going to be available. It was much more a question of would it ever be available? So that's a huge transition in the field. Because of the success of drugs like Luxturna and other genetic therapies for inherited retinal disease, as I mentioned earlier, there's a tremendous amount of interest in this part of medicine on the part of biotech and pharma companies. So there's a lot of investment in getting therapies which have been demonstrated to work in the lab into patients. This clinical development path. So we get inquiries from companies all the time asking us, what therapies have you shown that could work that we could buy from you so we could develop them and apply them to patients? So this is not self-serving. This is something I say in the clinic as well. If you really want to help move this forward, keep doing what FFB's been doing. FFB's been supporting research in the field, which generates proof of concept for these therapies, often in an academic setting, sometimes more and more now in collaboration with commercial entities via the RD fund. That's what's driving the field forward. And then eventually be willing to consider participating in these trials. I know we've talked about some of the challenges of being participants in clinical trials. We haven't talked about how much time they take. Take a lot of time on the part of the participants. But when a company develops or licenses a therapy and wants to bring it to clinical trial, one of the things they want to know is, can we enroll the clinical trial? Can we perform the trial, get an answer? Luckily, there's such a motivated community in the inherited retinal disease space. The answer is almost always yes. But what that depends on is the activity of people like Christine to do clinical trials and actively, which takes a lot of time away from other patient activities, and takes a lot of patients who are willing to consider participating in studies. So I feel like we're all doing the right things already. It's happening.
I agree. There's a lot of activity and there are multiple clinical trials and definitely frustrating, especially if you have one of the genetic mutations in a family member that doesn't seem to be right on the forefront. And so it's frustrating. I have a lot of patients in that ballpark who come into my clinic and, when are we going to have therapy for such and such genotype because it's not yet in the forefront. I think that also just reminds us that there are going to be non-genotype related therapies and, back to stem cell therapy. I actually wanted to correct something I said earlier where I was talking about the J-site and the reneuron trials as being stem cells. So those are the retinal progenitor cells trials that intravitreal injection for J-site and the subretinal reneuron sort of stem cell therapy, retinal progenitor cells. And then back to, what Astellas did many, many years ago with ABCA4 disease with Stargardt and AMD, those were stem cell derived RPE cells. So those types of trials where it's stem cell related therapy, those are also non-genotype specific and might be able to have much broader inclusion criteria. Again, with optogenetics and fields such as neuromodulation, which have much broader inclusion criteria and are not genotype specific, might end up being something that is worthwhile. And you have to start a debate. I have patients, for instance, with USH2A who don't have the exon 13, so they're not able to be in the ProQR study. And they keep asking me, should I wait for an USH2A augmentation gene therapy trial, or should I get into one of these RP trials because I have RP and I could theoretically be in a future J-site or reneuron type trial. So those are good questions, and you really have to kind of think about what you would like to be involved in. But and I know you're specifically asking about AAV trials, but there are different types of trials that will potentially be available, in a shorter period of time. Everything takes time, and I think involvement, investment, of course, being able to, and patients always ask me if I do happen to have, if I would like to invest, what do I do with it? What do I do with this? And probably the answer is donate to nonprofits like the Foundation Fighting Blindness, RPB, so groups that are actually supporting, or directly to a Mass Pioneer, so to a group that's actually working on your genotype, because funding certainly is an important part of being able to get things moving forward, or to the sponsor, that might be working on your particular genotype, so those are awkward things to say, but to be honest, that is important to be able to support, and also just to, I have patients who literally, go to those visions walks every year and try to help and be, with time, be able to donate their time to be able to be involved in helping to raise funding for research groups who are working on diseases that they have. And so I think that's kind of the long answer to what to do now, is just continue to support research and be involved and stay educated. And it is overall, it's an encouraging statement that there really is a lot, like Dr. Pierce said, there is a lot going on. It just, you have to unfortunately be patient with all of this because some things take some time.
Yeah, thank you for that answer. And I just want to sort of foot stomp that point. I believe that, the sort of CRISPR-Cas technology, as far as being able to be a tool that we could control, is less than 10 years old. Is that correct? Am I correct in that? And we've already seen it. We already see it in a person working, which is absolutely amazing. So I just want to let our attendees know that, in this biomedical field that we work in, we have hit an inflection point to where now we are sort of transiting from basic biology into the clinic to see if we can have some of these solutions in hand. And that's because we have some of the tools in hand now. So yeah, just continue to ask these questions, continue to push the scientific field and the medical field with your demand for solutions. And trust me, people are dedicating their lives in order to solve these problems, and they will meet the challenge. The next question we have is, if you have two sort of competing therapies for a particular, let's say, genetic mutation, how would you go about deciding which therapy to administer to a patient? And Dr. K, we'll start with you.
I'm sorry, I just clicked off for a second. Can you just repeat the question? I can't hear it and I can't see it.
Oh, I'm sorry about that. It's, for example, if someone has a particular genetic mutation, however, there's two potential therapies for it. Let's say one is a
Okay. How do you decide?
gene therapy
Gotcha.
versus
Okay, gotcha.
gene editing. How would you choose?
Great question. And certainly I've had this exact question with patients with CEP290, because even though it's a rare disease, and then we're even more specific looking at the Intron 26, we have 2 trials that they could theoretically get into right now. And I have one woman in particular, a patient of mine, who literally has had this phone call with me multiple times. And it's a difficult question to ask, and even harder to answer. So should she be in the ProQR antisense oligonucleotide therapy, or should she be in the CRISPR-Cas9 gene editing therapy? And so I do my best to give her the data from those 2. We have more data on the ProQR antisense oligonucleotide therapy. Now that we have that paper, that Steve Russell paper from 2018, I try to actually literally print out the paper and send it to patients so they can look at the data and have an educated decision. And I explain, most importantly, the modality of treatment and the risk. So that would be, for instance, an intravitreal injection with repeat therapy every 3 months or whatever it's going to be as it moves into the Illuminate, which is the phase 3, versus CRISPR-Cas9 is potentially a long-term expression therapy, potentially a one-time treatment, but it is more invasive. So meaning it is, correct me if I'm wrong, Dr. Pierce, but it's a subretinal injection, correct, with the vitrectomy. So very different modality of delivery here. So we're putting a patient through a surgery with a vitrectomy, doing a subretinal injection. So even though we're not injecting, it's not, well, it sort of is gene therapy, it's gene editing, but in any event, that is a much more invasive therapy. Patients have to kind of understand the risk-benefit scenario of the type of therapy. And then we have a little bit less data because that's earlier on, that's in the earlier phases of trials as compared to the antisense oligonucleotide. And I don't usually tell a patient what to do. I almost never tell a patient what to do because the patient has to make up their mind. So my role as an investigator and a clinician is to give them as much information as I can in terms that they would understand. And I am always impressed with my patients of how much they do want to know the actual gory details and see the actual papers. So I give them as much information as they want and do my best to explain it to them in the best terms I can. I even literally call Dr. Pierce's clinical coordinator and asked a few more details to get information. So I'm often making phone calls myself or ask Dr. Pierce as a curbside question, what do you think? So getting kind of questions from the people who are actually doing the trials to try to get their advice. But then again, I still don't answer the question. I let the patient make up their own mind with as much information as I can possibly provide them. And then I also say, here's the clinical coordinator contact for the Mass Eye and Ear group. You can talk to this lady, she's really good. So try to get her the correct contact info. And so she can continue to make up her own mind and ask those questions to the sites that are actually doing the trials.
Dr. Pierce, would you like to add anything to that?
No, I think Christine got it all right. They're difficult choices. You try to pull together to make a choice based on as much information or data as possible. And I think Christine is entirely correct. It's a personal choice for each individual.
Okay, we have about just sort of under 3 minutes left. We have one more question that's come in, and it's essentially, it's essentially this person is looking to maybe get in touch with other families who have children maybe with Nmnat1 mutation. Do you know of any sort of groups that have formed that they could essentially contact or reach out to?
Yeah, there's actually a foundation that was formed specifically around Nmnat1 associated disease. They are connected to, affiliated with it, at least by relationships with FFB, but the Gavin R. Stevens Foundation was set up by a family who have a child, Gavin Stevens, with Nmnat1 associated disease. And I'm not breaking any HIPAA privacy guidelines here because they have been quite public in advocating for research and raising support for research on this disease.
All right, thank you. So with just about 2 minutes left, I would like to sort of offer each of you a chance just to give any sort of closing thoughts or any last words that you'd like to tell the audience. So Dr. Pierce, we'll start with you, and then Dr. K, we'll close it out with you.
I think all I'd say is thank you very much for your very thoughtful, provocative questions. I hope the discussion we've had has been helpful.
Yeah, and I just want to thank you to all of you who are involved on this on a Saturday. I wish we were all here in person. It is different doing these conversations in a virtual setting. Am I on mute? Oh no, there I am. No, I'm on mute, but I've been doing lots of calls because of COVID on virtual. But the Foundation Fighting Blindness was excellent in being able to put this together amidst COVID and us not being able to travel. Everybody stay safe. I live in Florida. My state is not doing well right now as far as COVID. I had to go get the nasal swab myself twice in the last week because there was a potential exposure. Luckily, I'm negative, but I mean, that's a big deal right now with COVID. So all of you, please stay safe and let's hope we get through this and get a vaccine. But back to LCA, this is just a very exciting talk to have been invited to give. Again, I think that it might be the most exciting because there's so much going on in this particular field with LCA, with an FDA-approved therapy and so many trials going on. And so if your particular genotype is not in a clinical trial yet, I think it's like Dr. Pierce said, it's not a question of if, but it's now just a question of when, which is really exciting. So just stay involved in the Foundation Fighting Blindness. Make sure you've had genetic testing. If you haven't, if there's anyone who hasn't asked the question, these questions have come quite a ways in the last couple of years of me doing this. I remember a few years ago, our LCA talk was, how do I get genetic testing? What is genetic testing? Now these questions are, tell me about NMAT1 and tell me about CRX. I mean, it's incredible, you know, how much the information has been dispensed in the last few years and how educated patients and families are now. So I think that level of education and involvement is really moving forward the field. And so it's very exciting. I feel like I picked the right field for my life mission here because I really enjoy working with the patients and the families who have this, and it's an exciting time. And thanks again to the FFB for putting this together.
Yeah, and again, I'd like to thank you, Dr. K and Dr. Pierce, for your participation in the session. Also, I'd like to thank all the attendees and all your great and wonderful questions. If you have any more questions and they were not addressed during the session, you can please email us at info@fightingblindness.org. And with that, this concludes our session. Thank you again. You have a wonderful rest of your day. Take care.
Bye-bye.
Bye now.